Introduction. Nanotechnology has revolutionized various fields, and medical science is no exception. With the advent of medical nanotechnology, scientists and researchers have been able to explore new avenues for drug delivery, imaging, and therapeutics. Nanoparticles, in particular, have shown immense potential in enhancing drug efficacy, minimizing side effects, and enabling targeted therapies. In this script, we will delve into the intricacies of medical nanotechnology, focusing on different aspects such as ligand-conjugated nanoparticles, passive targeting, active targeting, intracellular drug delivery, surface modification strategies, multifunctional nanoparticles, controlled drug release mechanisms, biodistribution, pharmacokinetics, nanoparticle-based gene delivery and therapeutics, theranostic nanoparticles, nanotoxicology, and translational challenges in nanoparticle drug delivery. Through an in-depth exploration of these topics, we aim to provide a comprehensive understanding of this rapidly evolving field. Ligand-conjugated nanoparticles have emerged as a promising approach to achieve targeted drug delivery. These nanoparticles are functionalized with specific ligands that can bind to receptors overexpressed on the surfaces of target cells. By selectively binding to these receptors, ligand-conjugated nanoparticles can enhance the accumulation of therapeutic agents at the desired site, thereby improving their efficacy and reducing off-target effects. The design and optimization of ligand-conjugated nanoparticles require careful consideration of parameters such as ligand selection, surface modification techniques, and characterization methods to ensure efficient targeting and enhance therapeutic outcomes. Passive targeting is a phenomenon that takes advantage of the unique characteristics of tumor tissues, such as leaky vasculature and impaired lymphatic drainage. Nanoparticles can exploit these features to passively accumulate in tumor tissues through the enhanced permeability and retention (EPR) effect. This effect allows nanoparticles to preferentially accumulate within the tumor due to their small size, prolonged circulation time, and specific surface properties. The EPR effect has been extensively studied and utilized to improve the delivery of anti-cancer drugs, imaging agents, and other therapeutics. However, several factors can influence the efficacy of passive targeting, including nanoparticle size, surface charge, and shape. Thus, careful consideration of these parameters is crucial for successful passive targeting strategies. Active targeting involves the use of ligands, antibodies, or other targeting moieties to actively deliver therapeutic agents to specific cells or tissues. This approach capitalizes on the specific interactions between targeting molecules and their corresponding receptors on target cells. Active targeting offers several advantages over passive targeting, as it enables precise targeting and enhanced accumulation of therapeutic agents in the desired cells or tissues. Various strategies have been employed for active targeting, including the use of nanoparticles functionalized with specific ligands, antibodies, peptides, aptamers, or small molecules that can recognize and bind to target receptors. The rational design of active targeting strategies involves careful consideration of parameters such as ligand selection, surface modification techniques, and targeting molecule-receptor interactions. Successful active targeting approaches have been reported for various diseases, including cancer, cardiovascular disorders, and inflammatory conditions. Achieving effective drug delivery inside cells remains a significant challenge in drug development. Nanoparticles offer promising solutions for intracellular drug delivery, as they can efficiently overcome biological barriers and deliver therapeutic agents to specific intracellular compartments. The design of nanoparticle-based intracellular drug delivery systems involves considerations such as cellular uptake mechanisms, endosomal escape strategies, and intracellular trafficking. Nanoparticles can be engineered to enhance cellular uptake through receptor-mediated endocytosis, direct membrane penetration, or fusion with cellular membranes. Additionally, strategies such as pH-responsive or enzymatic degradation of nanoparticles can facilitate drug release within specific cellular compartments. Understanding the complex interactions between nanoparticles and cellular components is essential for the successful development of intracellular drug delivery systems. Surface modification plays a crucial role in tailoring the physicochemical properties of nanoparticles for specific applications. Nanoparticles can be functionalized with various ligands, polymers, or biomolecules to impart desired properties such as enhanced stability, prolonged circulation, improved biocompatibility, and targeting capabilities. Surface modification strategies include, but are not limited to, PE-gylation, covalent attachment of targeting ligands, incorporation of stealth polymers, or coating with biomimetic materials. These strategies can significantly influence the pharmacokinetics, biodistribution, and cellular interactions of nanoparticles, thereby impacting their overall therapeutic efficacy. Careful selection and optimization of surface modification techniques are essential for achieving desired functionalities without compromising the nanoparticle's stability and biocompatibility. Multifunctional nanoparticles possess a unique combination of properties that enable them to perform multiple tasks simultaneously. These nanoparticles can integrate therapeutic agents, imaging agents, targeting moieties, and stimuli-responsive elements within a single nanoscale platform. By incorporating multiple functionalities, multifunctional nanoparticles offer enhanced therapeutic outcomes, precise imaging capabilities, and the potential for personalized medicine. The rational design and engineering of multifunctional nanoparticles involve considerations such as the selection of appropriate materials, surface modification strategies, and characterization techniques. The development and utilization of multifunctional nanoparticles hold vast potential for revolutionizing diagnostics, therapeutics, and precision medicine. Controlling the release of therapeutic agents from nanoparticles is a critical aspect of drug delivery systems. Achieving controlled drug release ensures optimal drug concentrations at target sites, minimizes systemic toxicity, and prolongs therapeutic efficacy. Various mechanisms can be employed to facilitate controlled drug release, including stimuli-responsive systems, for example, pH, temperature, light, magnetic fields, biodegradable materials, or external trigger activation methods, for example, ultrasound, near-infrared irradiation. The selection of appropriate release mechanisms depends on factors such as the desired release kinetics, nanoparticle properties, and the specific requirements of the therapeutic agent. A deep understanding of these mechanisms is necessary for the development of efficient and precise controlled drug release systems. Nanoparticles' biodistribution and pharmacokinetics play a crucial role in determining their effectiveness as drug delivery systems. Understanding how nanoparticles interact with various biological barriers, such as the immune system, cellular uptake mechanisms, and circulation dynamics, is essential for optimizing their pharmacokinetic profiles. The physicochemical properties of nanoparticles, including size, shape, charge, and surface modifications, significantly influence their interactions with biological components, which, in turn, affect their biodistribution and clearance from the body. Studying the biodistribution and pharmacokinetics of nanoparticles provides critical insights into their therapeutic efficacy, toxicity, and potential interactions with other drugs. These findings contribute to the rational design and optimization of nanoparticle-based drug delivery systems. Gene delivery holds immense promise in the field of medicine, enabling targeted therapies and personalized medicine approaches. Nanoparticles offer several advantages for gene delivery due to their ability to protect nucleic acids from degradation, facilitate cellular uptake, and enhance intracellular delivery. The design of nanoparticle-based gene delivery systems involves considerations such as nucleic acid encapsulation, surface modifications, and release mechanisms. These systems can target specific cell types, facilitate intracellular trafficking, and control gene expression levels. The development of safe and efficient nanoparticle-based gene delivery systems requires careful optimization of parameters such as particle size, surface charge, biocompatibility, and gene delivery payloads. The advancements in nanoparticle-based gene delivery have the potential to revolutionize the treatment of genetic disorders, cancer, and other complex diseases. Theranostic nanoparticles combine therapeutic and diagnostic functionalities within a single nanoscale platform. These nanoparticles offer the unique capability of delivering therapeutic agents while simultaneously providing imaging capabilities for disease diagnosis, treatment monitoring, and visualization. The integration of therapeutic and diagnostic functionalities within a single nanoparticle allows for real-time monitoring of treatment efficacy, personalized medicine approaches, and enhanced patient care. The development of theranostic nanoparticles involves careful selection of imaging modalities, therapeutic agents, surface modifications, and release mechanisms. Additionally, understanding the interactions between nanoparticles and biological components is critical for achieving precise imaging and therapeutics. Theranostic nanoparticles hold immense potential in revolutionizing disease diagnosis, treatment, and monitoring. As with any emerging technology, the safety and potential toxicity of nanoparticles are a significant concern. Nanotoxicology focuses on studying the adverse effects of nanoparticles on biological systems and elucidating underlying mechanisms. The physicochemical properties of nanoparticles, such as size, shape, surface charge, and surface modifications, deeply influence their interactions with biological components and potential toxicity. Understanding the toxicological profiles of nanoparticles is crucial for their safe and effective use in medical applications. Nanotoxicology research encompasses various aspects, including cytotoxicity assessments, immune responses, genotoxicity, and long-term toxicological effects. The insights gained from nanotoxicology studies enable the development of safe and biocompatible nanoparticles for medical applications. Despite the remarkable advancements in nanoparticle-based drug delivery, several challenges still hinder their successful translation into clinical applications. Translational challenges encompass a wide range of aspects, including regulatory approvals, manufacturing scalability, stability, toxicity concerns, cost-effectiveness, and clinical trials. Understanding and addressing these challenges are crucial for bringing nanoparticle-based drug delivery systems from bench to bedside. Collaborations between researchers, clinicians, regulatory authorities, and industry stakeholders are essential for advancing nanoparticle technologies and overcoming translational hurdles. By identifying and addressing these challenges, we can facilitate the clinical implementation of nanoparticle-based therapies and improve patient outcomes. In conclusion, medical nanotechnology, particularly the utilization of nanoparticles, offers unprecedented opportunities in drug delivery, imaging, and therapeutics. The intricacies of ligand-conjugated nanoparticles, passive and active targeting, intracellular drug delivery, surface modification strategies, multifunctional nanoparticles, controlled drug release mechanisms, biodistribution, pharmacokinetics, gene delivery, theranostic nanoparticles, nanotoxicology, and translational challenges have been explored in this script. Each topic provides detailed insights into specific aspects of medical nanotechnology, highlighting the immense potential and complexities involved in harnessing nanoparticles for precise and effective treatment strategies. Continued research, innovation, and collaboration are crucial for unlocking the full potential of medical nanotechnology and translating it into tangible clinical benefits.